After studying about the protein synthesis, that is, how a protein or a polypeptide chain is formed from DNA, that is, the entire central dogma process involving replication, transcription and translation, we will now understand about how this protein containing amino acids and genes within carries out its expression. That is, how does the regulation of gene expression occurs. Before understanding the regulation, let us understand the term gene. Gene can be defined as a DNA segment in chromosome which acts as a heredity unit and controls the characters by transmitting it from parents to offspring. That is, the entire control of the heredity that takes place from parents to offspring is mainly carried out with this DNA segment which is present within the chromosome called gene. As studied previously, according to Mendelian theory, gene was termed as factor. That is, before describing the term gene, it was depicted as factor by Mendelian theory. Which was later called as gene. Now, gene exactly consists of four main regions. These regions are named as the first, promoter, second, initiation site, third, coding sequence, and fourth, termination site. Thus, these four regions together constitute a gene. This figure depicts the structure of a gene. As it can be seen from the figure, these four regions constitute a gene, where the first region, that is the region located at the initial end, is known as promoter, following it comes the initiation site, and at the end is termination site, and the sequences which are present within is known as coding sequence. Thus, this entire thing constitutes a single gene. Therefore, the location of promoter is at the one end of the gene to this region. RNA polymerase is attached, which helps during the RNA synthesis. Then the next site or the next region that comes is initiation site. This region is located next to promoter . From here, the mRNA synthesis begins. Then the third region, that is coding sequence, this region is located at the middle segment, that is located at the middle segment part of the gene. The main function of this is that all the nucleotides that are present are copied into the mRNA. Following it comes the last region, that is termination region. This region is located at the other end of the gene, that is exactly opposite to the first one, that is promoter gene. The main function of this region is that the mRNA synthesis gets stopped here. Now, after studying about the structure of gene, let us begin that how the genetic expression is being regulated. Let us have an overall idea about the regulation of gene expression. As we all know that in various organisms, different genes are responsible for the synthesis of different proteins. However, it is not necessary that all these proteins are needed at the same time. That is, different proteins that are formed by different genes are regulated according to various functions at different times. And therefore, for example, the enzymes or the proteins that are needed during the childhood time is not necessary that those will also be needed during the adulthood time, and vice versa. On this basis, it is not so that the genes within an organism are present only in certain age, because genes that regulates the protein synthesis occurs within all organisms throughout lifetime. And so, on this basis, it is not always necessary that all the proteins code at all the times throughout the life of an organism. There is always a mechanism involved which helps in the regulation of such a gene expression. But the mechanism in both prokaryotes and eukaryotes is entirely different. That is very obvious that within prokaryotes, that generally includes bacteria, have comparatively simple mechanism for regulation of such gene expression. But on the contrary, the eukaryotes, which have a complex body structure and organization, have comparatively complex mechanism, that is, exhibit comparatively complex mechanism for such a regulation. And so, it was easy for scientists to study about the mechanism of prokaryotes. On this basis, various scientists proposed different theories, out of which the best one was from the French scientist named Jacob and Monod, who discovered such a mechanism in Escherichia coli or E. coli bacteria, for which they were even awarded the Nobel Prize. In this, they found that for an expression of gene, that is gene expression, there may be one or a few more genes involved. That is, in order to exhibit a particular expression that is regulated by gene, it's not necessary that only single gene is involved, because in some situation even more than one gene can also involve to exhibit such an expression. Thus, in this manner, the gene whose product is required may be expressed, and such a gene is called structural gene. And so, structural gene is very essential because it is a gene which helps in the expression to obtain the desired product. On the contrary, which is entirely opposite to it, there is another gene known as repressor gene. It is a gene that can block the gene, that is operator gene, that produces the product, and therefore no product is formed. And therefore, repressor gene, in other terms, represses the gene so that its functioning does not occur, and RNA polymerase cannot form mRNA. Thus, structural gene helps in enhancing the expression. On the contrary, repressor gene represses the function. As a result of repressor gene, since no mRNA is formed, therefore transcription is also prevented. And since transcription is prevented, in this manner no protein synthesis can occur. However, there are a few situations in which the repressor gene can also become deactivated. How? If an inducer gene that act as substrate in presence of hormone which can also be said as precursor. So, when such inducer acts as a substrate, it can bind to the repressor protein and therefore opens up the operator gene. That is, after the inducer gets bind to the repressor gene, then onwards the function of repressor protein would get deactivated, which would help in the activation of operator gene, and thereby the transcription can occur. And so the RNA polymerase can also form mRNA, and this RNA polymerase thereby bound to the promoter site, which helps in the transcription and formation of mRNA. This figure depicts the role of the hormone as an inducer substance. As it can be seen from the figure, the repressor protein, which is represented by R, in its presence blocks the operator, that is O, as a result of which the structural gene becomes inactive and the RNA polymerase gets blocked, with the help of which or due to which the transcription does not occur. But in the second situation, when the repressor gets bound to the inducer, that is in the presence of inducer, the repressor protein becomes deactivated and this blockage gets lifted up, and as a result of which the operator gene becomes active and results into the formation of mRNA through activated transcription. Here is the presence of hormone which helps in the integrator that activates the RNA that binds to the receptor to form mRNA through transcription.